This is the lecture for Chapter 7, Control of Microbial Growth, Part 2. In order for cells to grow, they need a source of nutrients, and with those nutrients, they will perform catabolic reactions and anabolic reactions. And all of that relates to energy, releasing energy, storing energy, and all of that is used to build cellular structures and to perform cellular processes such as cell division. So all of this constitutes microbial growth. And in order for cells to grow, they need functional enzymes. So they need these enzymes so that they can catalyze all the reactions that are required by their metabolism. Controlling microbial growth, in other words, preventing microbes from growing, is important in many different settings. It's important in a laboratory setting to prevent contamination of experiments. It's important in healthcare settings to prevent infection or to cure infections. And it's also important in the food industry to prevent spoilage. There are different methods of controlling microbial growth, but all of these methods will have a particular mode of action. And most of the time, these modes of action are either to have an effect on the cell membrane, have an effect on the enzymes, or to have an effect on the DNA. And all of these modes of action will at least prevent the cells from metabolizing and or kill the cells. With the different methods of control, they are divided into two groups: physical methods of control and chemical methods of control. The first group of methods of control that I want to go through are the physical methods of control. And physical methods of control relate to the environment that the microbes will be in. Within this group, temperature is the first aspect that I want to talk about, and within temperature, I want to talk about increasing temperature or heating the environment. All microbes are dependent on their enzymes. They're dependent on having functional enzymes so that they can metabolize, and if they can metabolize, then they can grow and replicate. This is a diagram that gives the relationship between temperature and enzyme activity. And every enzyme has an optimal temperature where it works at its fastest rate. It also has a temperature range, and that indicates all the temperatures where there is at least some activity. If you do not want the microbes to grow, you need to take them out of their temperature range. And temperature has an effect on protein structure. As you increase temperature, the hydrogen bonds that are in the protein, in the enzyme, will break, and that protein, that enzyme, will denature. So it will lose its three-dimensional shape. If you increase temperature only a little bit and then cool it back down to the optimal temperature, this process is reversible, so that those proteins will be able to renature and become functional enzymes again. But if you increase the temperature extremely, then you will permanently denature the protein, denature the enzyme, and when you denature it, it loses its three-dimensional shape, so it loses its function. When using heat as a physical method of control, there are two different types of heat that can be used: dry heat and moist heat. Dry heat usually involves some sort of flame, like a Bunsen burner. Moist heat involves water, and that could be boiling water or steam. But both types of heat are capable of sterilizing substances. And you know in lab that we use the Bunsen burner to sterilize the loops before inoculating different types of media. Moist heat is used in the lab to sterilize media. And remember, the definition for sterilization is that there are no microbes present, or all microbes, all forms of the microbes have been inactivated. Boiling water, if you boil water that is contaminated with vegetative microbes, they will die in about 2 minutes. Endospores are more resistant to boiling, so you would need to boil for about 20 minutes. But boiling water or dry heat will be able to kill the endospores, so both methods are capable of sterilization. When comparing the two methods, moist heat is actually more efficient because that sterilization can occur at 100 degrees Celsius. And again, that is the temperature where water boils. Flames are also capable of sterilization, but the temperature of a flame is about 1,500 degrees Celsius. So when you look at the temperature, moist heat is more effective at lower heats than dry heat. And the reason for this is just the amount of molecules in the substance. When you are using dry heat, you're looking at the air. So it's the air, the molecules in the air that are bouncing against the bacteria, leading to the death of the bacteria. When you have moist heat, you have more particles. Water molecules are involved, so you have more particles that are bouncing against the microbes, causing damage to the microbes. So that is why moist heat is more efficient than dry heat. An example of using heat as a physical method of control is the autoclave. And an autoclave uses steam under pressure to sterilize different things. In a laboratory setting, media, so the media that we use to culture cells, is placed in an autoclave so that it is sterilized. And remember, sterilized means that there are no active forms of microbes in any form. No vegetative microbes, no endospores. So in a laboratory setting, media will be autoclaved. And also in a medical setting, you have all the surgical equipment. So all the surgical equipment will be autoclaved, and in a dental setting, all the dental hygiene equipment will be sterilized in an autoclave. The second example of how heat can be used as a physical method of control is pasteurization. And pasteurization was developed by Pasteur, who was the father of microbiology, very involved in the golden age of microbiology. And he is the one who proved biogenesis. And one of the things he needed to work on was to prevent the spoilage of food. So preventing the spoilage of food, he found that if you heat the food, that will kill many of the microbes, so that will prolong the shelf life of the food. So in pasteurization, not all of the microbes are killed. So pasteurization is actually a type of sanitization. So the milk that is pasteurized that you see in the grocery store, it still contains some microbes, but they have been reduced to a safer level, and will prolong the shelf life of the milk. But you should realize that milk, even if you buy it from the grocery store, you never open it, it will still spoil in the refrigerator because it is still contaminated with some microbes, and over time they will grow and spoil the milk. There are some types of pasteurization, like high temperature, short time pasteurization, that can be used to sterilize. But most of the milk that you find, especially the milk in the refrigerated section, has undergone regular pasteurization and it still contains microbes. Another way of using temperature as a physical method of control is to reduce temperatures. So using cooling to reduce the growth of microbes is very common, especially with food preservation. And all of us do this at home with a refrigerator or freezer. So refrigerating the food, freezing the food is a method of microbial control. And the way this works is, again, by affecting the enzyme activity. Every enzyme has that optimal temperature, and when you reduce the temperature from that optimal temperature, you get a gradual decrease in activity. And the reason you get that gradual decrease is because reducing temperatures reduces the ability of the enzyme to perform induced fit. So reducing temperatures is not going to denature the proteins. It does not denature proteins. They still retain their normal shape. They're just unable to form that induced fit, those hydrogen bonds to the substrate, so they are unable to stress the covalent bonds in the substrate so they cannot facilitate the reaction. The thing with cooling is that it is bacteriostatic. Remember, bacteriostatic means that it inhibits the replication of the microbes, but it does not cool them. So what happens is when you put contaminated or food that has been exposed to microbes in the freezer, when you freeze it, the microbes stop growing. But once you take that food out and warm it up to room temperature, then this is reversible. So if you warm up that food, those microbes, to room temperature, their enzymes will become functional again and they will grow. So cooling is bacteriostatic. It will slow the microbes down or stop their replication, but it will not kill them. So if you warm them back up to within their temperature range, they will start replicating again. Another physical method of control is pH. And pH affects enzyme activity because every enzyme has an optimal pH where it functions at its fastest rate. And if you change the pH, either increasing the pH or decreasing the pH, that will have an effect on the enzyme's activity. And the reason it has an effect on enzyme activity is because it interferes with hydrogen bonding. So it interferes with the ability of that protein to hold its secondary structure and, of course, its tertiary structure, because hydrogen bonding is also involved in holding that tertiary structure. So increasing pH or decreasing pH, either one, will lead to denaturing the protein. And similar to heating, if there's just a tiny change in pH, that can be reversible. The protein can renature. But if you have an extreme increase or decrease in pH, then it is a permanent denaturation. One example of using pH to prevent food spoilage is pickling. So adding vinegar to different vegetables, it decreases the pH of the environment, and bacteria, most bacteria prefer a neutral pH, and so that takes them out of their pH range and prevents the bacteria from growing and spoiling the vegetables. Another physical method of control is osmotic pressure. And this refers to the type of environment that the bacteria is in and the relative solute concentration. Bacteria are able to survive in isotonic environments and hypotonic environments, but if you place them in a hypertonic environment, they will experience osmosis. So water will osmose out of the cell because the bacterial cell is hypotonic compared to the environment. And that will cause the plasma membrane to shrink as the cell is losing all that water, a process called plasmolysis. And because of the loss of water, the bacteria will no longer be able to metabolize. They won't have enough water for the reactions. And also the proteins will denature because this will have an effect on that hydrogen bonding. So the way to create a hypertonic environment is to add solute. Remember, the solutes are all the things that are dissolved in the solvent in the water. And we use this method to prevent spoilage of foods. There are two types of solutes that we can use. One solute is sugar. And when we take fruit and add a high amount of sugar to create a hypertonic environment, we create jelly or jam, or the old-fashioned word, preserves. Because what were they doing? They were preserving the fruit from spoilage. Another type of solute that we can add instead of sugar would be salt. And that is a common way to preserve meat, is to put the meat in a high salt environment, either in a brine or to rub salt over it, and that creates a hypertonic environment which inhibits the growth of bacteria which would otherwise spoil the meat. Another physical method of control is desiccation. And when you say something is desiccated, that means the water has been removed, or the majority of water has been removed. And again, this will have an effect on the bacterial cells, because if you remove the water, they won't have water available for the catabolic and anabolic reactions. And also this has an effect on protein structure. And desiccation can be used as a method of preserving food. So drying. When fruits and vegetables are dried, put in a dehydrator, the water is removed, and that can preserve them, prevent them from spoiling. Also, a related type of desiccation is freeze drying. So these bananas have been freeze dried. That is drying under cold temperatures, which preserves the structure compared to desiccated or air-dried bananas. And one of the things I came upon when I was looking for pictures of desiccated food to prevent spoilage, I actually found a picture of taxidermy. So apparently taxidermy now can be used with, or freeze drying can be used with taxidermy. And if you think about it, the whole purpose of taxidermy is to prevent the structure of the animal from decaying. And decay is caused by microbes that are breaking down the structure of the animal. So again, it's a method of microbial control, is to get rid of those microbes, prevent those microbes from breaking down or decomposing the structure of the animal. So removing the water, freeze drying the structure, will prevent the microbes from growing and using that structure as nutrients. Another physical method of control is filtration. And filtration is a little bit different than the other physical methods of control, because this will not directly affect the membrane or the enzymes or the DNA of the microbes. In filtration, you are physically removing the microbes from the environment. So basically with filtration, you have a filter which has pores of a certain size, and those pores need to be smaller than the microbes. So when you pass a substance through the filter, the microbes are going to be prevented from flowing through. So the microbes remain on one side of the filter, so they are physically removed from that substance. And filtration can be used in air, so those HEPA filters, those are filters that will filter the air. They will trap the microbes, or most of the microbes, so that as the air flows through, many of those microbes are removed. Now, filtering air can just sanitize it. You cannot sterilize air, but you can sanitize the air. You can reduce the number of microbes to safe levels. Filtration can also be used on liquids, and you pour the liquid through a filter, and the filter, if the pores in the filter are small enough, then the liquid that comes through is microbe-free. And actually, this can be used to sterilize liquids, so they can actually be sterilized. And in lab, when scientists are culturing animal cells, like human cells, they cannot heat the media in an autoclave, because that will destroy important proteins for the eukaryotic cells, for the animal cells. So the only way to sterilize the media for animal cells is to filter it. The last physical method of control is radiation. And to understand how radiation affects microbes, it's important to understand the electromagnetic spectrum. And this is the electromagnetic spectrum. It organizes different types of radiation by wavelength. And so the way this one is organized, the radio waves have the longest wavelength, that's about 1 kilometer, whereas the gamma rays have the shortest wavelength, which is on the order of nanometers. And the important thing is to know how wavelength relates to energy. It's actually an inverse relationship. So wavelength is increasing in this direction. As it increases in that direction, the amount of energy the radiation has is going to decrease. So energy increases in that direction. So the longer the wavelength, the less energy. The shorter the wavelength, the more energy. And sometimes I like to think about it visually. A long wavelength means that it takes the particle a long way to go to its crest, and a long time to go to its trough. And so this is a long wavelength. Whereas a type of radiation that has a short wavelength, it's going to go up and down very quickly. So up and down very quickly, so it has a shorter wavelength, it has more energy. With the electromagnetic spectrum, it's important to know the different types of radiation that it has. And usually the easiest place to start on the electromagnetic spectrum is with visible light, and this is where the visible light is. When you separate white light, all those photons by their wavelength, you get the rainbow. And it's easy to remember the order of the rainbow with the acronym Roy G. Biv. And this diagram is a little bit opposite. So it starts Roy, red, orange, yellow, and G is green, and Biv is blue. Oops, I wrote backwards. Blue, indigo, violet. So that's how you can remember the order of the colors. And the important thing to remember is that red has the longest wavelength. Violet has the shortest wavelength within that group, within the visible light. So red has the least energy. Violet has the most energy. So once you get that orientation, it's pretty easy to figure out the rest of the electromagnetic spectrum. So if we start on the red side and go longer in terms of wavelength, so what has a longer wavelength than red? It would be infrared. And infrared has a longer wavelength, less energy. It is not damaging to cells because infrared cameras are taking pictures of radiant heat, just the heat that organisms release. And that doesn't have a lot of energy. It's not damaging to cells. Even longer than infrared are microwaves, and this is referring to the microwaves coming from outer space. And so microwaves have a long wavelength. They're not very damaging. And really long, radio waves, those wavelengths can be a kilometer, almost the length of a mile. Those have very little energy and they don't damage cells. If we look at the other side of the electromagnetic spectrum, so we start out from the violet side, and now the wavelengths are getting shorter, so the energy is increasing. So shorter than violet is ultraviolet. So ultraviolet is even shorter than violet. It has more energy, and ultraviolet can damage cells. You know that if humans are exposed to too much UV light, it can damage our cells, damage our DNA, and it can lead to cancer. Even shorter than UV are X-rays, and you should know that X-rays can cause more damage to cells, and even shorter than X-rays are gamma rays, and gamma rays are associated with radiation poisoning. So this is the type of radiation that has the most energy. It has the shortest wavelength, so it can cause the most damage to cells. There are two types of radiation that can be used as physical methods of control. And the first type of radiation is called non-ionizing radiation. And the ionizing part of this description relates to the ionization of water. So water, if it interacts with radiation that has enough energy, can be ionized, broken in half to form hydrogen ions and hydroxide ions. So non-ionizing radiation does not have enough energy to break water apart, so it cannot ionize the water into the two different types of ions. An example of non-ionizing radiation is ultraviolet light. So ultraviolet light is non-ionizing radiation, and the effect that it can have on a cell is it can damage the DNA. And you know this because you should know that people who expose themselves to ultraviolet light run the risk of developing skin cancer. So cancer, of course, is caused by damaged mutations to DNA. So radiation will have an effect on the microbe's DNA. And non-ionizing radiation can only affect microbes that are on a surface. So it does not have enough energy to penetrate into substances. So because of this, non-ionizing radiation cannot sterilize. So it cannot sterilize by itself. The best it can do is disinfect, because remember, endospores are resistant to radiation. But non-ionizing radiation, it can have an effect on vegetative cells. There are many examples of using non-ionizing radiation, or UV light, as a physical method of control. One example is in tissue culture. So some of the hoods or benches where tissue culture is done, there is a UV light up in the top. So in between using it, the UV light is turned on to disinfect the surface of the bench and where the scientist was working. Also, UV light can be used in air filters. So this is an air filter where there is a HEPA filter filtering the air, removing some of the microbes, and those microbes that are too small to be removed, the UV light can help disinfect that air. Also, UV lights are being used in water sanitation to help disinfect or sanitize the water. They're also starting to use UV lights in hospital rooms. So again, this can be used only on surfaces. So the UV light is only going to affect the microbes that were left on the surfaces, and this can help sanitize a hospital room. And the last interesting example of UV light are personal equipment that can be used to sanitize things like your cell phone or glasses or anything that can fit in this small container. So there are a lot of different uses for UV light as a non-ionizing radiation to control microbial growth. But remember, it cannot sterilize and it can only disinfect at the best, but only on surfaces because UV light cannot penetrate. The second type of radiation that can be used as a physical method of control is ionizing radiation. And this is called ionizing radiation because it does have enough energy to ionize the water molecules. And since it has more energy than non-ionizing radiation, that means it has a shorter wavelength than non-ionizing radiation. So in the electromagnetic spectrum, ultraviolet was the non-ionizing radiation. So ionizing must have a shorter wavelength, and that indicates that ionizing radiation are the X-rays and the gamma rays. So they have very short wavelengths and they have more energy. Ionizing radiation also affects the DNA, but because it has so much energy, it is able to penetrate into substances. And you should be able to remember that because what are X-rays used for? They're used to take pictures of internal structures like teeth and bones. Because ionizing radiation has so much energy, has such a short wavelength, and can penetrate into substances, it can be used to sterilize. And they are starting to use ionizing radiation, specifically gamma radiation, to sterilize different substances. And when they do this, they build a facility that has the radioactive material, and then they run whatever it is they want to be exposed to the gamma radiation through this facility so it can be exposed to the gamma rays. And what happens is you have your substance and the gamma rays pass through the substance and they damage the DNA of the cells that are inside the substance. So gamma radiation uses, there are quite a few. Some of them have to do with damaging the DNA to prevent a process from occurring, like sprouting of different types of vegetables. So if the DNA is damaged, then those vegetables will not produce sprouts. Also, it can be used to kill insects or insect eggs that may have contaminated the food. Shelf life extension, basically that's preventing spoilage. So they can run the meat, the fish, through the gamma radiation, and that will kill many of the bacteria that are on the surface, which will extend the shelf life. Pathogen reduction, that again is also killing microbes, and these would be the microbes that could spread disease through the humans who eat that food. And the last thing, the quarantine. This is used to kill insects or microbes that could spread diseases to other plants, native plants. So ionizing radiation involves gamma rays and X-rays, and it is so powerful that it is possible to use it for sterilization. In addition to the physical methods of control, there are also the chemical methods of control. And this involves adding chemicals, molecules, atoms to an environment to kill or inhibit the growth of the microbes. And one way to determine the effectiveness of different chemicals is to perform a disk diffusion assay. And the disk diffusion assay involves creating a lawn of bacteria on a plate and then adding on top of the plate discs, filter paper discs, that have been soaked in the various chemicals. Then the plate is incubated for 24 to 48 hours, and as the chemical diffuses through the agar, if it is able to inhibit the growth of bacteria, you will observe a zone of inhibition. And in terms of chemicals, the relative size of the zone of inhibition will indicate the relative effectiveness. So in this case with E. coli, the largest zone of inhibition is with the chlorine, which means it is much more effective than the hexachlorophene here that has no zone of inhibition. Now I want to go through several different types of chemical agents that are used as chemical methods of control. And one of the things I want to point out is that often these different chemical agents are referred to as disinfectants. And that is because they do have the ability to disinfect something, and that means to kill all the vegetative microbes but not the endospores. But they have to be used appropriately in order to actually disinfect a surface. So often these disinfectants are actually sanitizing surfaces. But another thing to point out is that by themselves they are unable to sterilize anything. So the best they can do is disinfect. The first type of chemical agent that I want to talk about are the phenols. And phenols were first discovered by Lister. He was a surgeon during the golden age of microbiology, and he decided to put a phenol compound on the surgical wounds of patients to prevent infection. And that was very effective at that time, but phenols have an effect on the plasma membranes of microbes, and also because eukaryotic cells like human cells also have a plasma membrane, the phenols are actually caustic to human tissue. They can actually also damage human tissue. So now we do not use phenols on living tissue, we only use it on surfaces, inanimate surfaces. And an example of a phenol we use today is Lysol. So Lysol is mainly a phenol component. So what Lister was doing is he was basically putting Lysol on the surgical wounds. So you can imagine what effect that would have had on the human tissue. The next group of chemical agents that can be used as a method of control are the halogens. And halogens are actually a column on the periodic table of the elements. So that's where the group halogens can be found. And the effect the halogens have on the microbes is they can affect the protein structure. So they can denature enzymes which will affect the metabolism of the microbes. One example of a halogen is iodine, and iodine can be used for antisepsis. So it can be used on tissue, human tissue, to prevent infection from a surgical wound. Another example of a halogen is chlorine, and chlorine is most often associated with water. So chlorine can be used to disinfect water, and it's often used in sewage treatment plants where chlorine is added to the water that has been filtered, and that is used to disinfect that water. And chlorine also is often used in pools. So that is a good way for you to remember that it can be used to disinfect water. Another group are the heavy metals. And the heavy metals are also found on the periodic table of the elements. So these are the heavy metals. And within the heavy metals, two of the most common that are used for antimicrobial control are copper and silver. And both of these have an effect on the proteins. So they will denature the proteins. And an example of silver, silver is often used as a treatment for burns. And it is very good for treating burns, especially if the patient is exposed to antibiotic-resistant bacteria. And recently, they have been incorporating silver into the creams, of course, that are used on the burns, but also into bandages and even gloves that can be put on burned hands. So not that I am promoting any sort of product because I do not work in healthcare, so I don't use these, but I kind of like it that in the name they have Ag, which is the symbol for silver. Alcohols are another group that can act as a chemical method of control, and alcohols actually have two targets. They're able to disrupt membranes and also denature proteins. So alcohols are very toxic to bacteria, to cells in general. And alcohols are often used for degerming, using those alcohol swabs to swab the skin before an injection. And also alcohol is the main component in hand sanitizer. The next group are the surface active agents. And the surface active agents are more commonly known as soap or detergent. And the effect that the surface active agents have, or the soap has on the microbes, is to disrupt the phospholipid bilayer. So this is an example of going through how soap works against COVID-19. And this is the soap molecule here. It has a part of it binds with the water and a part binds with the lipids. So when soap comes in contact with an enveloped virus, and remember the envelope is composed of a phospholipid bilayer, the soap molecules will disrupt the envelope. And the envelope is important for attachment and penetration. So if the envelope is disrupted, then the virus cannot infect the cells. And so hand washing with soap is very, very important and very effective at protecting individuals from enveloped viruses. The last chemical agent are peroxygens. And peroxygens are formed when oxygen gas interacts with water to form oxides. And remember, those oxides are capable of damaging protein, DNA, and also lipids. And that's not just on the bacteria, but also on eukaryotic cells. And the most common peroxygen that can be used as a chemical method of control is hydrogen peroxide. And again, that is formed when oxygen interacts with water. Sometimes when people get a scrape, you could add hydrogen peroxide to that scrape to kill any microbes that have entered into the tissue. And if you do add hydrogen peroxide to a scrape, you will often see the formation of bubbles. And the formation of bubbles is caused by catalase. And catalase is the enzyme that can break down the hydrogen peroxide back into oxygen gas and water. So what the catalase does is it detoxifies the hydrogen peroxide. Now, you probably should not use hydrogen peroxide on tissue because most of these bubbles that are being produced are being caused by the human catalase that is detoxifying the hydrogen peroxide. So of course this hydrogen peroxide would be killing the bacteria, whatever bacteria have entered into the wound, but it is also damaging the human cells. So hydrogen peroxide is also toxic to human cells. So it's much better to use the hydrogen peroxide on inanimate surfaces rather than on tissue. The last slide is just a reminder to go over the relative resistance of the different types of microbes. So this is sort of a review of the structures of the microbes and what may make them more resistant or less resistant to different methods of control. And also you may want to look at your microbes to memorize list to get an idea of which microbes may be more difficult to kill or less difficult to kill. So just as a reminder, prions are the most resistant, may be completely resistant to any sort of sanitization, disinfection, or sterilization method. Then looking at just the bacteria, endospores will be the most resistant form of bacteria, and the Clostridium species and the Bacillus species can form endospores. When comparing gram-negatives to gram-positives, the gram-negatives will be more resistant because of the structure of their cell wall, having that outer membrane which can prevent a lot of chemicals from entering into the cell. Mycobacteria are kind of a special group of bacteria. They cause tuberculosis and leprosy, and because they have mycolic acid and lots of waxes in their cell wall, that makes them more resistant to the regular types of bacteria. Looking at protozoa, there are those that have cysts and those that are vegetative, the trophozoites. And the whole purpose of the cyst is to be a protective form. So of course, cysts are more resistant than trophozoites. The fungi are just right there in the middle. And then the last group to compare are the viruses. And you are comparing what is their outer covering. The naked viruses just have a capsid. The enveloped viruses have that phospholipid bilayer. And it is relatively easy to disrupt a phospholipid bilayer compared to a protein capsid. So it is much easier to inactivate enveloped viruses compared to naked viruses. And the whole purpose of that outer covering is to perform attachment and penetration. So if you disrupt that outer covering, you're going to prevent the virus from being able to attach and penetrate into a host cell.